Welcome. First, very basic question is what is titration in the pharmaceutical industry? Answer is titration is a quantitative analytical technique used to determine the concentration of a substance in a sample solution by reacting it with a standardized solution that is titrant of a known concentration, or in other words, titration is used to find the unknown concentration of an analyte by the known concentration of a solution when a indicator is present. This one is very commonly asked question. Question is what are the commonly used titration analysis techniques in the pharmaceutical industry? So answer is acid base titration, complexometric titration, redox titration and precipitation titration are the commonly used titration analysis techniques in the pharmaceutical industry. Next question, how is endpoint determination achieved in titration? Here are the 5 important techniques of endpoint determination. First technique is by using indicators. Here color change signals the completion of reaction. Second method is by using pH meters. Here we measure the pH shift at endpoint. Third method is by using potentiometric sensors. Here we detect abrupt change in potential. Fourth method is by using conductivity meters. Here we monitor the change in conductivity. And fifth method is by titration curves. What factors should be considered when selecting a titration method for pharmaceutical analysis? Here are the 5 factors that should be considered when selecting a titration method. These are: first, analyte nature; second, titrant characteristics; third, reaction kinetics; fourth, required sensitivity; and fifth, interfering substances. What is Karl-Fischer titration and its significance in pharmaceutical analysis? Karl-Fischer titration is a specialized titration method used to determine the water content in pharmaceutical samples, especially in drug formulations where precise control of water content is critical for stability and efficacy. What is principle of Karl-Fischer titration method of analysis? Karl-Fischer titration involves the reaction between iodine and sulfur dioxide with water in the sample. The reaction takes place in the presence of a suitable solvent and a base. The endpoint of the titration is determined by detecting the consumption of iodine, which indicates the complete reaction with water in the sample. What procedure is followed during routine titration method by endpoint determination by color indicator? Step 1. Pour a measured amount of the unknown solution, that is the analyte, into a flask. Then fill a burette with solution of known concentration, that is the titrant. Step 2. Slowly let the titrant drip from the burette into the analyte in the flask. Step 3. Watch the color of the analyte. When the titration is finished, the color of solution usually changes. This change tells you that you have reached the end point of the titration. How is the accuracy of titration results ensured in pharmaceutical analysis? Accuracy of titration results is ensured by following 5 ways. First, calibration of titrant solution. Second, precise sample preparation. Third, careful sample handling. Fourth, reproducible titrations. And fifth, method validation. Briefly explain about precipitation titration method. Precipitation titration is a method where precipitate is formed through the reaction between analyte and the titrant. The endpoint is reached when the precipitate forms completely, indicating the completion of the reaction. What is complexometric method for titration? In complexometric titration, a complex is formed when the reaction reaches its endpoint. This type of titration is more precise than precipitation titration because there is no risk of errors caused by co-precipitation. Briefly explain about redox method of titration. Redox titration involves a transfer of electrons between the used analyte and the titrant. These kind of titrations are named after the reagent that is used in, are as follows. First, permanganate titrations. Second, dichromate titrations. Third, iodometric titrations or iodometric titrations. What is titration curve? A titration curve shows how the pH of a solution changes as we add more of a titrant. On the graph, the horizontal axis, that is x coordinate, shows how much titrant we have added, while the vertical axis, that is y coordinate, shows the concentration of the substance we are analyzing. For acid-base titrations, the curve mainly tells us about the strength of the acid or base being tested. What are some challenges associated with titration in pharmaceutical analysis? Some commonly faced challenges are requirement of skilled personnel, second, sample interference, third, time consumption, fourth, equipment maintenance, and fifth, indicator selection. How does automation enhance titration in the pharmaceutical industry? Automation provides efficiency improvement, error reduction, it gives high throughput analysis, provides consistency and data management. Last and important question, which guidelines are commonly referred for titration in the pharmaceutical industry? Here are the list of 5 important guidelines that are commonly referred. It includes USP, that is United States Pharmacopoeia, EP, that is European Pharmacopoeia, BP, British Pharmacopoeia, and ICH and ISO guidelines. Don't forget to like, comment and share. Subscribe to channel for more videos related to our pharmaceutical industry.